Okay, close enough. I'd like to remind you that when you participate, you get 3 credits. So that's not the only reason I'd like you to. I'd like your feedback. But in each class that we have this semester, I'll award 3 credits to people who participate. Now, if you look in your assignments tab in Canvas, you should have 3. One is a worksheet on Introduction to Biology. Another is a worksheet, Methods of Scientific Inquiry, which is what we're going to be talking about today. And the third is quiz. Now, this quiz is unusual because, first of all, it's not graded. Secondly, it's a dry run. Canvas is now giving us a choice of using old quizzes or new quizzes. And we're strongly encouraged to use new quizzes because the old quizzes will be phased out. So I'm trying to use new quizzes, and it's quite different from my end. So I need to get feedback from you, how you receive the quiz and how easy it is for you to answer the questions. The other reason for this, and the reason that it's not graded, I want to give you an idea of the kinds of questions I asked and to work within the time limit. And in this quiz, you'll be given 2 attempts, and that will be the case in all quizzes. And then on, this is due on Monday before noon. And then we have class at 12:30 on Monday, and we'll discuss the quiz and any problems that you have with it. So any questions about what's coming up in terms of assignments? All right, you're a quiet group. Do you see the methods of scientific inquiry?
Yes.
Yep.
Okay. That's the topic for today. How do scientists investigate the natural world? What methods are used? Well, there are many different methods, and we'll review some of those today. But scientific investigation requires logic, a certain amount of analytical skill, and very important, perseverance, because usually best laid plans don't work out, and you have to try over and over, be very persistent to actually follow a line of investigation. And scientists are, aside from being scientists, they're ordinary people. So they live in the same world, have the same constraints, same distractions as non-scientists do, the exception being that scientists are trying to do science. You've probably all seen this diagram or something resembling it, which gives the prototypical summary of the scientific method. It's a straight line process. You make an observation. Based on that observation, you form a hypothesis. Then you test that hypothesis by doing experiments. And depending on how the experiments turn out, you develop a theory to explain the observations. Now, if the experiments do not give results consistent with the hypothesis, you go back and change the hypothesis. If, on the other hand, you do multiple experiments, some of them support the hypothesis and some don't, then you have no basis for a theory. So again, you go back to the hypothesis and make adjustments. So is this the way science is actually done in the real world? Rarely. It almost never works in such a straightforward manner. Now, there are two types of research, curiosity-driven and applied. Curiosity-driven is based on, I wonder what would happen if, and you can come up with anything you like, crazy things like I mixed red paint with green. What would happen if I did? What would happen if I put vitamin pills on my plants or poured sand in my gearbox? Or any number of different questions. The question can be as wild as you'd like, as long as there is a way to address it. Now, in contrast to curiosity-driven investigation, there is what's called applied research. Applied research has a practical application in mind before you even start. And it's an attempt to apply scientific methods to specific everyday problems. Now, this is the straight line representation of the scientific method. But this is trying to represent the way it actually works. So you'll notice that all the boxes are here, making observations, asking questions, doing experiments, thinking critically, communicating information. But notice that there's no straight line order here. In fact, all of these boxes are connected to each other by arrows, and all the arrows are double-headed, that is, they point in both directions. So that means you could start an investigation with any of these. You could start by thinking critically and going from there. You could start by having a communication, for example, online or by talking to somebody. You might be inspired to start an investigation. So, and by starting at a different point doesn't mean you won't do everything in all the boxes. It just means that you won't do them in the prescribed order. So does this make sense? I'm telling you the same thing in both of these slides. Here you have the traditional way that the scientific method is usually presented. And here you have a more realistic way of representing what actually happens.
Makes sense to me.
Okay, that was precious. Good. Well, keep letting me know whether things are making sense to you or not, and especially if they're not, we'll discuss it further. So, is there a single scientific method? No. Here are the elements of scientific inquiry repeated again in a list, but I've added here testing reproducibility. You do an investigation or a set of experiments, and you get a result. The question then is, can you do the same thing over again and get the same result? So, can you reproduce your own results by you? and then equally important by others. So if you communicate your results, you remember communication is one of the pillars of an investigation. You communicate your results either verbally or in print. And anyone who is interested can try to reproduce your results by doing exactly the procedures that you described. If others can reproduce your results, now it becomes more powerful, because it means not only does it work in your hands, other people can follow your instructions and get the same result. Here's an everyday example of observation and hypothesis testing. It's very simple. You get up in the morning, turn the switch, the light doesn't go on. So you can make a hypothesis. You can make several different hypotheses. The bulb is out, the power is out, or the breaker is tripped. So to test the first hypothesis, you replace the bulb. If the new bulb works, answer is yes, and you're done. On the other hand, you replace the bulb, still doesn't light. You check to see if other appliances on the same circuit, if they work. And if they do, that gives you that answer. If they don't, then you go to your third hypothesis, check the breaker switch, and see if that works. And if it does, then everything's fine. If it doesn't, none of your hypotheses worked, and something else is going on. For example, maybe the power company has shut off everything in your area. But anyway, this is a very simplistic but realistic way that we all go about making observations and testing hypotheses in our everyday lives. Now, different fields of investigation have different methods. Scientific method, there's an engineering method, mathematicians have a method, there are methods in law. So if we just compare scientific method with engineering method, there's a lot of commonality. So science asks a question, engineers define a problem. In both cases, background research is done. A scientist will construct a hypothesis, whereas an engineer will specify requirements. And you can go on down, matching up the colors, and see that in both cases, results are obtained, and then they're communicated. So the point here, we asked a question earlier, is there just one method of doing investigation? And the answer is no, every field has its own method of investigation. And the scientific method, which is used by scientists, has many variations. Okay, making observations and asking questions. Questions arise as a result of observations, and they can be addressed by either observational science, discovery-based, or experimental science, which is hypothesis testing, or in many cases, both. So let's look a little more carefully at these. Developing hypotheses and conducting investigation. A hypothesis is a tentative explanation or a possible answer to a question. So we have experimental science and observational science. Experimental science investigations are based on controlled hypothesis testing. Underline controlled. What do we mean by a control? A control is a constant. So throughout the experiment, it does not change. So, for example, an experiment in agriculture might be to examine the effect of fertilizer. Well, the control for such an experiment would be no fertilizer. Similarly, in medicine, if you were looking for the effect of a drug on a certain condition, the control would be patients that received no drug. So you're in experimental science, you're always comparing a treatment with a control. In other words, fertilizer versus no fertilizer, drug versus no drug. In observational science, investigations may or may not be testing a hypothesis. So for example, a geologist looks at rock formations, not necessarily doing experiments on them, but just observing what they look like. Political science. Usually political scientists do not do experiments because it almost always involves humans. So what is their database? It's historical precedent, usually. That is observational science. And as I said before, some questions can be addressed through using both observational and experimental. Okay, look at this for a little bit. What do you think is shown here?
Grass.
Grass with and without fertilizer?
Yes. Okay, well, that was Nick
Yes, sir.
and Fiona, I think. Okay. I'm just making notes of who is speaking up. Yeah, this is fertilizer versus no fertilizer. If we go back to the previous slide here, example, agriculture, effect of fertilizer. So the plants on the right were not fertilized, those on the left were, and you can see an obvious difference in growth. So that's an experiment. An experiment is a controlled test. In this case, the control is no fertilizer, and the treatment is fertilizer. So it's a controlled test. One variable should be changed at a time. And in any experiment, there is an independent variable and a dependent one. The independent variable is changed by the experimenter. The dependent variable depends on the independent variable. So that's illustrated on the right here at the top, independent versus dependent variable. So in this case, we're testing fertilizer. And the independent variable is treatment with fertilizer. The dependent variable is the response to that. And the response will be the plant. Growth, or the number of leaves or number of fruits, any parameter that you wish to choose to measure response of the plant. The results are always plotted on a graph which has an x-axis and a y-axis. The x-axis is always the independent variable. In this case, the amount of fertilizer or the composition of fertilizer, but the independent variable will be, has to do with the fertilizer. The response is the plant, and that is the dependent variable. It's always represented on the y-axis. So remember this, it will come up again. So let's look at some examples of observational or experimental science. A scientist goes to Alaska 3 years in a row, counts the number of polar bears found in a particular 5 square mile region, and reports that. Is that observational or experimental?
Observational.
Okay, that was Brianna. Yes, you're right. It's observational. There's no experiment involved there. Scientists just looking and counting. Very often, observational research involves counting. A scientist determines the DNA sequence of the gene that is responsible for cystic fibrosis. Observational or experimental?
Is that an
Observational.
experimental?
Okay, 2 people responded. Say your name, please.
Ramon?
Ramon, okay. And you said observational?
No, experimental.
You said experimental.
I said observational.
Esteban said observational. Okay, so we have one is experimental and one is observational. Anyone else have a thought?
I thought it was experimental as well.
You thought it was experimental, Brianna. Okay. Why do you think it's experimental?
Because they're trying to find the sequence of the DNA.
Uh-huh. And what's the treatment and what's the control?
I just assumed they would like change the DNA sequence slightly to see which one would cause cystic fibrosis and which one wouldn't.
That would be an experiment if you change the DNA sequence, but that's not what the statement says. It says it determines the DNA sequence of a gene. So nothing changes here. Nothing is being compared. It's just the DNA sequence of a particular piece of DNA. So those of you who said it's observational, you're right. You're just observing the sequence. You could think of it as counting. You're counting the nucleotides as you go down the chain of DNA. So that's kind of a tricky one. It sounds experimental, but when you think about it, it's observational. Okay, the next one, a scientist determines the rate of growth of plants given different doses of a new fertilizer. Some of the plants are given no fertilizer. What's that?
Experimental.
Experimental.
Uh-huh.
Experimental.
Yeah, you're all right. It's experimental. That's a fairly easy one. Okay, next one. Scientists take a submarine to an area of the ocean floor and look for types of organisms that live there.
Observational.
Observational.
Uh-huh. Yeah, they're just counting and categorizing. The last one, scientists test the growth rate of a newly discovered bacterium on different types of food source.
Observational.
Experimental.
Observational.
Okay, we've got both. Those of you that think it's observational, why?
They're just looking for the types of organisms. That's all. They're just observing which ones live there.
Okay, and those of you who think it's experimental, why?
Because it
So they're
states that the
testing.
scientists are testing it.
Say again.
The sentence states that the scientists are testing the growth. So in sense, they're experimenting on it.
Yeah, and so what do you see as the variable then in this statement?
I'm sorry, come again, my thing cut out.
What do you see as the variable in an experiment? If this is an experiment, we have to have a control and a variable. What's the variable here?
Would that be the bacterium?
No, it just indicates one bacterium, a newly discovered bacterium.
Oh.
What else might vary?
I think the variable may be the rate of growth, and maybe the control is no food source versus all the others having different types.
Okay, you're on the right track. The variable is the food sources, different types. So, for example, you could have a medium on which bacteria grow, and you might vary the sugar type in each medium. So one might be glucose, one might be sucrose, one might be lactose, and so on. And the scientist is going to measure the growth of one bacterium on these different types of sugar sources. So the variables here are the sugar sources. And what would be the control then?
No food source.
Yeah, you're right. That would be a control, but on no food source, there would be no growth. So for this experiment, you'd have to have something other than no food.
Would that be the bacterium?
Okay, this is getting a little complicated, so let me just explain. In this case, the scientist has to make a decision on what the control is going to be. So in order to do the experiment, the bacteria have to grow in all cases, so you have to give them some nutrition. So you choose a medium as the control. So for example, you could take a medium with glucose in it, say that's the control, and then you're going to compare it with a number of other sugar sources. So is this making sense?
Yes.
How you sort out how to do an experiment given the limited amount of information we have here. You choose a control condition, you choose a bunch of variables to compare with that, and then you inoculate them all with the same bacterium and measure growth. And there's any number of ways to measure growth of bacteria that we won't talk about, but there are standard methods of measuring bacterial growth. Okay, let's proceed. Proposing a hypothesis. A hypothesis is a possible answer or a tentative explanation for what you have observed. It's different from a theory. Theory is a broad general set of principles that can make predictions. A hypothesis does not make predictions. Hypotheses reflect past experiments with similar questions. Multiple hypotheses should be proposed whenever possible. Remember, in the case of the light being out, we had several different hypotheses for what might be the problem. Now, importantly, in yellow here, hypotheses must be testable and they must be falsifiable. Now, when we go further, we'll see that theories have to satisfy both those criteria as well. But in order for it to be a hypothesis, there must be a way to test it, and there must be a way to prove that it's wrong, if indeed it is wrong. Hypotheses can be eliminated, but can never be confirmed with absolute certainty. So there is no absolute truth in science. So let's compare science and pseudoscience. And this is really important for you to think about, especially now with so many conspiracy theories circulating on social media, so much misinformation and misunderstanding about science is going on. And much of this can be classified as pseudoscience. So let's just compare science with pseudoscience and think, in going through this, think about COVID-19, the coronavirus, all the controversies that are going on right now. So in the case of science, it changes with new evidence. Pseudoscience, there are fixed ideas. If you follow the news, you'll know that the recommendations for how to deal with the coronavirus change on a fairly regular basis. The changes are usually made by the CDC, the Centers for Disease Control. And why does the CDC make changes? Because there's new evidence. On the other hand, there are a lot of people who have fixed ideas about coronavirus or about vaccination. And they're not looking for new evidence, they're looking for support for their fixed ideas. And that's the next point. Science considers all the data. Pseudoscience focuses only on data that support the foregone conclusion. Science starts with an observation and ends with a conclusion. Pseudoscience does the opposite. Starts with a conclusion, a fixed idea, and then looks for evidence to support it. In science, the results have to be repeatable. Pseudoscience, they're not. Science invites criticism. Pseudoscience resists it. And science is driven by the desire to understand the phenomenon. So for example, in the case of COVID-19, we want to understand so we can use the best measures to combat it. Pseudoscience is driven by cultural or commercial or ideological goals, not necessarily aimed at solving the problem, but proving a point. So I'm sure you've all been exposed to science and pseudoscience in the real world. Any thoughts about this? Okay, no thoughts, we'll go ahead and compare a hypothesis with a theory. And this is going to come up again for you, so you'll probably want to be referring back to this when you do your worksheet. So the definition of a hypothesis is a suggested explanation for something that's observed. So it makes a prediction. A theory, on the other hand, is well substantiated. So it's a unifying explanation for a set of proven hypotheses. So there's no clear line of demarcation between a hypothesis and a theory. But if a hypothesis is tested over and over and over again, it can become a theory. If the testing always comes to the same conclusion. So a hypothesis is based on a suggestion. Theory is based on evidence that has been verified by repeated testing and by wide scientific consensus. Now you'll note the next two, testable and falsifiable, are common to both hypothesis and theory. If it's not testable or falsifiable, then it falls into a different category. And I'll talk about that in a little bit. Well substantiated, hypothesis no, a theory yes. Hypothesis is based on limited data, theory is based on a wide set of data tested under various different circumstances. A hypothesis is specific, based on a very specific observation. A theory is general. It's based on enough tests and experiments to draw a conclusion that you can make predictions based on the results. The purpose of a hypothesis is to present an uncertain possibility that can be explored further. The purpose of a theory is to explain a large set of observations that are consistently made by multiple investigators. So you'll want to think about these differences, and you'll use this information certainly in your worksheet, maybe in upcoming tests. Any questions? Now, if it's not a hypothesis, it's not a theory, it might be an assumption. How is a hypothesis different from an assumption? A hypothesis is not accepted as true. It's rather just one possibility, and it can be falsified. That is, it's testable. An assumption is not accepted as true. Excuse me, let me start over. An assumption is taken for granted. It's believed to be true based on no evidence. It's not testable. It doesn't need to be tested because it's accepted to be true from the outset. So we have a hypothesis, we have a theory, and we have an assumption. They're all different. Uses of the term theory. Theory is used commonly in everyday conversation. It's also used in science in a different way. So in day-to-day speech, theory often means a guess or an unsubstantiated idea. So you might hear someone say, I have a theory that gremlins are hiding my car keys. That is not a theory. It could be stated as a hypothesis, but even that is a stretch. But the point to be made here is that when you hear the word theory used in the media, for example, theory usually doesn't mean theory in the scientific sense. It means more that it's an assumption or a hypothesis. Now, as we've been saying, in science, the word theory has a very specific meaning, refers to a well-substantiated or proven hypothesis. Whereas a hypothesis is a suggestion, a theory is something that's substantiated by a lot of evidence. I think one of your worksheets deals with this and asks you to describe the difference in your own words. Thinking critically. You need to identify and articulate problems. The question you ask is critical. The words you use to ask it are critical. Then you gather relevant information, do investigations, and organize and analyze the information. Now, this is a very important part of the scientific method, how you display or present the data that you've collected. So I'd like to contrast two different sets of data here at the bottom right. In the first case, we have three different columns of numbers. And in the other case, we have a graph. Now, with those three different columns of numbers, what can you conclude? Is it possible to draw any conclusion just by looking at them?
I don't think so.
Yeah, you're right. You can sit and look at that all day and no conclusion jumps out at you. On the other hand, look at the graph on the right. What can you conclude from that? Oh, part of it is obscured. Let me move this. Now you can see bears, whales, and dolphins. Can you draw any conclusions from that?
You can see that the dolphin population has been dropping
Yeah.
year by year.
Uh-huh. And you saw that immediately, right? Within a few seconds of looking at that graph, you saw immediately that the dolphins are disappearing.
Right, and then the bears are increasing in population.
And you saw that immediately. Now, contrast that these data that are plotted on this graph could have been presented as three columns, in which case you couldn't have made any sense of it. But because it's displayed on a graph, you look at it and you immediately can draw conclusions. So the point to take home here is that you collect numbers. The numbers usually don't mean anything until you present them in some kind of a way that allows you to see trends. And very often, statistics are applied to sets of numbers. So for example, if you looked at the numbers in 2018, did a statistical analysis, you'd probably see no differences at all. But if you applied statistics in 2022, there are big differences, probably statistically significant. Okay, communicating information. This is one of the tenets of the scientific method. Once you've acquired new information, it is your responsibility to let others know. And there are different ways to do this. One is to write it down and publish it according to the format in whatever book or journal you want to publish in. You're probably all familiar with the magazines Science and Nature. There's another series of journals called PLOS, the Public Library of Science, whereas many journals require you to pay to have your work published. The Public Library of Science publishes free. So you can publish, that is mandatory for most types of investigation, and people in your field will read whatever it is you publish. Another way to communicate is to attend scientific meetings, conferences, or scientists bump into each other and to non-scientists all the time. And just by talking, discussing results is a very effective way of communicating information. So any questions about anything I've said on the slides? If not, I want to show this video. Oh, before going to that, here are some thought problems. I'm not going to go over these, but consider this a study guide. Read each of these on your own time and think about them. And the thoughts that you have will probably be useful for you in subsequent tests or worksheets. So as I said, I'm not going to talk about these now, but rather show you a video.
The scientific method is often used in the study of biology. It's how scientists gather new ideas and analyze information. While the scientific method is often presented in a linear manner, in practice, it rarely happens this way. The process tends to be more circular and often double backs on itself. Let's take a look at an example to better understand how the scientific method works. Let's say you decide to spend the day at the beach. After a couple hours, you notice your skin is bright red from sunburn. You know you applied sunscreen, so you're curious as to why this has happened. Let's use a simplified version of the scientific method to help figure this out. The first thing you might do is begin to make observations. You know you applied SPF 5 sunscreen. You don't typically get sunburns, but you are also outside for a longer period than usual. You remember reading that when sunscreens reach their expiration date, they tend to lose their potency and may not prevent sunburn as well. Maybe that's why you're sunburned. You decide to set up an experiment to test this hypothesis. In order to do this, you need to test the contribution of a single variable, in this case the expiration data, to the observation, your sunburn. So to test this hypothesis, you collect a number of sunscreen lotions, all with the same SPF, but different dates of expiration. You apply these to different areas of your arm and proceed out to the beach. 2 hours later, you notice that all the areas are showing signs of sunburn. So you now know that expired sunscreen isn't the reason you got sunburn, and your original hypothesis is rejected. You also remember hearing that sunscreens differ in their sun protection factor, or SPF values. Maybe your sunscreen wasn't a high enough SPF to block UV light, resulting in your sunburn. You decide to test this hypothesis by testing the contribution of a single variable, in this case the SPF value, to the observation, your sunburn. You collect a variety of similar sunscreens with different SPF values and apply them to your arm. After 2 hours at the beach, you analyze the results on your experiment and reach a conclusion. Sunscreens with higher SPFs do indeed block more UV rays than sunscreens with lower SPFs. It's important to recognize that the scientific method is not used by all disciplines in exactly the same way. Instead, it represents a process of thinking by which scientists analyze an observation, identify variables that may contribute to the observation, and then devise a form of experiment to test their hypotheses. Often the results of the experiment direct the researcher in a new direction, make them look at a problem in a new way.
So I've got my essay written and I've been working on it for about a week. So now,
That was an advertisement, in case you were wondering, which you don't need to listen to. Okay, that's it for today. do you have any questions or comments about anything today or anything that's coming up? The worksheets or the quiz, which will be on Monday.
I think I'm
I have
good.
a question about a worksheet.
Uh-huh.
How do you edit the Microsoft Word?
How to edit Microsoft Word.
It let me
Anybody?
know that the section was locked. That's why I'm asking if is there any way you can unlock it or what I could do?
Has anybody else had a problem with the worksheet?
No, but I've tried it in a different format.
You switched it to what format?
PDF, and I use I don't know if it's a Chrome extension, but it's connected to my Google Drive called Doc Hub, and I always just convert PDFs to there, and then I can edit them and then re-export it with my edits to my Google Drive for submission.
That was helpful. Thank you.
Okay. Would it be better if I post documents as PDFs instead of Word?
Yeah, like I, for example, use
Yeah.
GoodNotes on the iPad, and it's actually only allows me to edit it as a PDF and not a document.
Okay, all right. Well, that's good to know. I'll use PDFs from here on. So Fiona, your problem is solved? Or at least you have a direction to go.
Yes. Thank you.
Okay, thanks, Precious, for that.
No problem.
Anything else? Okay. Well, the quiz, ordinarily, if it were an ordinary quiz, it wouldn't be available until the day of the quiz. I've made this one available starting today, so you can take the quiz anytime you like. You get 2 attempts at it, and each attempt is 15 minutes. So you can do it anytime between now and Monday at noon. Monday at noon is when it locks. Okay. I'm going to end the session, and I will see you all on Monday.
Thanks, Professor.
Thank you, Professor.
Thank you.
Thank you.
All right.
Monday.
Yeah.
See you Monday. Bye.